Hey, how's it going, everyone? Mr. Binder here. This is the second part of Chapter 7 that I would like to discuss with you. The first part was more specifically the process that occurred to make protein. And again, this is a cellular process. A cell will take necessary steps in order to synthesize a protein. Transcription, translation, those were the two big stages that needed to occur in order for a successful synthesis of a polypeptide or eventually a protein. Again, what we're going to be discussing in this part is regulating gene expression. All right, proteins are made from genes. Genes are located in your DNA. Right? DNA is obviously located inside of the nucleus, very important. So when a cell needs a protein, it is going to look to the DNA. The DNA is the instruction manual. That's where proteins are, that's where the recipe books or recipes for proteins are found. Inside the nucleus, found on the DNA, there are segments of DNA that are called genes, and these genes code for a specific protein. Not all genes need to be turned on, and a lot of genes actually in specific cells will be turned off. So regulating gene expression will be the topic we're going to be discussing in this first part here. It's essentially the cell's mechanism to increase or decrease protein production. So again, a cell doesn't need genes on all the time, and a cell isn't continuously producing protein. A cell will turn genes on when protein is obviously needed. An example here would be an enzyme. Cells need enzymes. Enzymes are protein. In order for a cell to function properly, it obviously needs to synthesize a protein. Again, an enzyme would be an example of a protein. Okay? So when an enzyme actually needs to function inside of a cell, or when an enzyme is needed for a specific chemical reaction that's occurring inside of the cell will turn the gene on and then start making that enzyme. Now there's a couple things here that I'd like to talk about. Genes can be expressed or regulated in a couple different ways here. There's 4 ways: transcription, post-transcription, translation, and post-translation. This is going to be a test question. You will need to know all 4 of these different ways that genes are expressed or regulated in a cell. And we'll talk about each one of them. Transcriptional regulation. What we're going to be talking about is how genes are activated and how genes are silenced. Okay, activating gene transcription. Well, this is basically transcription factors are proteins that help to initiate transcription of a gene found on our DNA. All right, what 2 ways I'm going to be talking about here is DNA acetylation, okay, and what's known as a protein remodeling complex. All right, so this is a specific way through transcriptional regulation where genes can become activated. All right, DNA inside the nucleus is known as chromatin. All right, we know that chromatin condenses into chromosomes just before mitosis occurs, but as a cell is an interface, the DNA will not be in chromosomal form. It'll actually be in the chromatin form. Again, having said that, these proteins or transcriptional factors will again identify specific regions on the gene, attach to them, and basically wait for other processes to occur in order for transcription. If a cell needs a gene to be turned on, there's 2 ways. A protein remodeling complex can loosen the promoter region, right, which is probably wrapped around a histone. Okay? That's the first way. There's another way. It's called DNA acetylation. Right? Enzymes can add acetyl groups to histones. Adding acetyl groups to histones can expose the promoter region as well. Now a histone is like a spool, and imagine it's a spool and the thread is like the DNA. The DNA wraps itself around that spool. That's what a histone is. A histone is a protein. Well, if the promoter region is somehow wrapped around that histone, it needs to be exposed in order for gene transcription to occur. And these 2 ways, again, protein remodeling complex and acetyl groups added to histones, are a way in which a gene can be activated inside of a cell. Now, when genes don't need to be used for transcription, they can be turned off. And the way that this is, one of the ways inside of a cell that this can occur is called DNA methylation. So a specific enzyme will add methyl groups to the cytosines of a specific gene, and basically what happens here is it will become silenced or inhibits transcription. The genes will be placed on hold until they're needed again, and then acetylation or DNA acetylation or transcription factors, a lot of things come into play here to activate an actual gene for transcription. Here again we just see silencing, DNA methylation, methyl groups added to the cytosine, the nucleotides there. Post-transcriptional regulation is the next topic that we're going to be discussing. The first one was transcriptional regulation. It's basically turning genes on or off inside of a cell in order to make an mRNA transcript. Post-transcriptional means that the mRNA transcript is now synthesized. Okay, there's a couple things that I would like to discuss here. RBPs, rate of mRNA degradation, hormones being used to extend the lifespan of specific transcripts, and then microRNA. Right? So again, RBPs, those RNA binding proteins, they're used in pre-mRNA processing for alternative splicing and polyadenylation. RBPs are very important because they will regulate gene expression or the mRNA itself. Right? The rate of mRNA degradation, right? What happens here when the mRNA is transcribed and it gets sent out into the cytoplasm, there could be some sort of masking protein basically telling the cell that, hey, we don't need this mRNA transcript. Translation will not occur. That mRNA transcript that you see here, because of the masking protein which is inside the cell, that masking protein will then basically trick the cell or tell the cell that, hey, this mRNA transcript doesn't need to be translated, so the cell will degrade that mRNA transcript. Now on the flip side, if specific mRNA transcripts need to be extended inside of the cell and more protein needs to be made, a signal or a specific hormone will be used to extend the lifespan of specific mRNA molecules. Casein is the example here. Casein is a protein found in the mammary gland, okay, in women's breasts. Here we see that if casein, or if a female needs to produce more milk, right, because she needs to feed her baby, casein will obviously need to, or the mRNA transcript that will produce casein, which is the protein, will need to be extended in the cell. Prolactin is a hormone. Prolactin will then send a signal to the cell saying that, hey, this mRNA transcript needs to be extended. Do not degrade the mRNA transcript, so translation can continue to occur and casein can again continue to be synthesized. Okay? The last part is microRNA. MicroRNAs, they're produced by this dicer protein, and what microRNAs do, and they can block protein production. Now another cool thing about microRNAs is that they are being studied for early cancer detection. Translational regulation. Now, we've seen that the mRNA transcript was synthesized. Okay. Some modifications occurred to that mRNA transcript. It's now being sent out into the cytoplasm, okay, where translation will occur. Now, during translation, a couple things will occur. One of the modifications to the mRNA transcript was this poly A tail here. Okay, this poly A tail. The length of the poly A tail will determine how long the mRNA transcript will be able to become translated outside of the cell. A longer poly A tail will probably see the mRNA transcript out in the cytoplasm being translated a little bit longer if it was a shorter tail. Okay. Enzymes will degrade mRNA after it has been translated. Deadenylase is the enzyme that will remove the poly A tail. And again, the exonuclease will degrade the mRNA after the poly A tail has been removed. Now again, the length of the poly A tail will determine how long the mRNA transcript will be translated in the cytoplasm. Post-translational regulation. This is the last part of how genes can be expressed in a cell. A couple things here. The Golgi body or the Golgi apparatus, that is the finishing or the shipping and receiving of the cell. That's where proteins are sent for activation. Okay, or inactivation. It just depends on what the cell needs at this point in time. Proteolysis is removal of specific sections of polypeptides to activate or inactivate a specific protein. Okay. Again, chemical modifications can be made to a polypeptide. Phosphorylation, we've seen this in multiple chapters where phosphate groups are being added to specific molecules to activate them or put them on hold. Now, once a protein is tagged, or a protein, I guess we could talk about enzymes here. If an enzyme loses its function in a cell, because an enzyme is a protein, if it loses its function in the cell, a cell will degrade it. Now, it needs to be tagged for degradation. All right, basically quality control in a shop, right? You take a look at a part, it's not good. Well, what are you going to do with it? You put a sticker on it saying, hey, there's something wrong here. Well, that's what a cell does to protein when it's not needed anymore, or when it has become damaged. So what does a cell do? It tags this specific protein with ubiquitin. Right? Once it's tagged with ubiquitin, we see that a specific enzyme, a proteasome, will come and degrade that protein. All right, these are the 4 ways that genes can be regulated inside of a cell. All right, you need to know each 4 because all 4 will be on a test. Okay? The next thing that I do want to talk about is genetic mutations. Genetic mutations. There's positive and negative genetic mutations. Obviously, positive mutations are due to natural selection, evolution. Negative, cancer, which could eventually lead to death. We will talk about 2 small scale, single base pairs, and large scale, okay, multiple bases or whole genes. Mutations, they are, if they are bad, and if mutations occur inside of the cell, and again, this could be, DNA wasn't replicated properly, or we are introduced to maybe some chemicals or radiation, things like that, and again, something happened along the DNA that our cell couldn't find. What is cancer? There's the regulatory mechanisms here to actually fix our DNA. That's not found inside of the cell. And the reason why is because the mutations have occurred in the genetic code. All right, any mutation or anything that's responsible for a mutation inside of the cell, obviously affecting DNA, is called a mutagen. All right? And again, there are a variety of mutagens out in the world. Okay? The environment, of course, radiation from the sun, smoking, different chemicals, things that were introduced, maybe even food. Mutations are passed on to daughter cells because the regulatory mechanisms here didn't catch the mutation found in the DNA. Well, what happens here when a normal cell, when its DNA is mutated? Well, and it goes unnoticed. Well, that cell will continue to divide. That division will then form what's known as a tumor. Now, benign tumors are not cancer, they're just mass growths of cells proliferating. Right? Malignant would be your example of cancer, and malignant has the ability to metastasize throughout the body and start affecting other organs or other places in the body. And cancer, or if we're talking about DNA here, specifically speaking about what cancer is, mutated genes, and these mutated genes would be oncogenes. They're called oncogenes. Now these oncogenes, when they're turned on, they will cause undifferentiated cell division, and we would see a mass grow here, and that would be known as a tumor. Again, some causes of genetic mutations. Again, environment. The regulatory mechanisms in the cell couldn't catch a specific mutation or an error in the DNA replication. Chemicals, radiation, again, UVA, UVB rays. There's a lot of things that can cause DNA not to be replicated properly. These are some examples of chemical mutagens. Nitrous acid, again, nitrous acid is a product of a chemical reaction that occurs in our body through metabolism. Like sodium nitrate is the I guess molecule to blame here. Sodium nitrate is found in a lot of food. Well, it's additive, a lot of food additives. Okay, smoked meats and things like that. When you eat these, or you eat this food and you ingest sodium nitrate, your body will metabolize that and convert it to nitrous acid. Well, nitrous acid can modify individual nucleotides here, right? Which isn't good. These nucleotides will obviously resemble other base pairs. It will confuse the DNA or the enzymes that are responsible for DNA replication. Another one's ethidium bromide. It basically mimics a nucleotide base, and it can insert itself right into our own DNA. Aflatoxin, right, is produced by specific molds and fungi. All right, this is found in peanut butter and corn. Again, its aflatoxin has been approved by the FDA, and what aflatoxin can do is cause mutations, specific mutations to this gene P53 here. P53 is a tumor suppressor gene. Okay, it's kind of like the angel gene. Now, when there's a mutation specifically to this gene, well, our cells aren't going to be able to identify or specific mutations that occur in the cell. Right? And again, the question that you got to ask yourself is this aflatoxin producing mutations in the P53 gene, is that a direct result to cancer? What we are going to talk about, small-scale mutations. The reason why these are important is when the cell is making protein, it needs to get the information from the DNA. Well, when there's a mutation in the genetic code, specifically a gene, that is going to change the outcome of the polypeptide. We're going to take a look at a couple different things here. Missense, nonsense, silent, and frame shift mutations. All of these are examples of small nucleotide or single nucleotide polymorphisms, all right, or point mutations. All right. The first one is the missense mutation. This is a change in a single base. All of them will be. Okay. Basically, what these are substitution mutations. So again, you got 5 players on the basketball court. You want to make a substitution. One guy goes out, another guy comes in. There's still 5 people on the court. Okay, so missense is a substitution mutation. All right. So what happens in a missense mutation? Well, the mutated base pair CG, it should have been a T and an A. Well, again, if that happens on a specific gene, that gene is then transcribed into mRNA, and that mRNA is translated into a polypeptide. What we see is a different amino acid. That amino acid will affect the function of that polypeptide. Right? So again, a missense mutation here, the result of a missense mutation is a different amino acid in the polypeptide sequence. And again, this can have a direct result of protein function. The next one is a nonsense mutation. Again, we see a substitution mutation here, right? Where there was a C and a G, there's now an A and a T. Well, what happens there when it's transcribed to mRNA and then translated to a polypeptide? Well, a nonsense mutation codes for a premature stop codon. Well, that premature stop codon is, well, a stop codon is found in the mRNA to stop the whole translation process. Now, if that UGA or whatever codon it is, stop codon, there's 3 of them. The one here, the example here is UGA. If that UGA code, okay, is before the actual stop code because of a mutation that had occurred in the DNA, well, the polypeptide won't be finished. Okay, there's a premature termination of translation. The polypeptide will not be finished, and the protein will then not function properly. A silent mutation, again, this is where we see a substitution mutation occurring. A substitution mutation, so where there was a T and an A, there's now a C and a G, same thing. But again, a silent mutation means that there is no change in the polypeptide sequence. Reason being is because we've seen that amino acids have multiple codons, which is cool because, you know, if there's a silent mutation, right, what ended up happening is through translation, the amino acid still, or the polypeptide or the amino acid sequence was still the same. Okay, so the protein's not altered. All right, even though there was a substitution mutation here, we saw that the CG is substituted for the T and the A. Okay, even though there was a substitution mutation, the end result is still the same. Okay, the protein's not changed, and again, we still see that protein as functioning properly. Frame shift mutation is a little different. Okay, first we saw missense, change in a single amino acid. Nonsense, a premature stop codon. Silent mutation, nothing changed as a result of the mutation. Okay, those were all substitution mutations. We're taking a look at frame shift mutations. These are also known as addition or deletion mutations. Okay? Basically what happens here is we're adding another nucleotide base, okay, into the DNA. Now this could be an example as in hockey, you know, when you got too many men on the ice, right? We got too many men on the ice, there's a penalty. Well, the penalty here obviously is we're going to get a series of amino acids that are altered because of this frame shift. So the whole reading frame of the mRNA transcript changes. And as the machinery, so the rRNA and the tRNA come together and start reading the mRNA transcript and start translating, when it finally gets to that part where there was where there was an addition or deletion of the actual DNA here, okay, we see a change in the amino acid sequence. Okay? This will obviously alter the protein. The protein will not function properly. Okay, so frame shift mutation, think addition of a nucleotide base pair or the deletion of a nucleotide base pair. The addition or deletion of these nucleotide base pairs will change the whole reading frame of the mRNA transcript. When that occurs, the amino acids will then obviously be totally different. Okay, so we see multiple amino acids here that are different as a result of a frame shift mutation. Those were all small scale or single nucleotide polymorphisms. That will be on the test. I would like you guys to know each of those. I might give you an example of DNA, and I might say, oh man, mutation occurred. Tell me what type of mutation it is. So you would actually have to go through the transcription process, the translation process, and then again tell me what type of mutation that is. Okay? These next series of slides here, the large-scale mutations, this will actually be probably matching or some sort of multiple choice question. I'm not going to ask you too much on this. They're pretty straightforward in terms of these of the mutation that we're talking about. But large-scale mutations are multiple nucleotides, not just a single base pair. It's multiple nucleotides or entire genes. Not only that, but also regions of chromosomes. Amplification, okay, this is gene duplication here. All right, so again we see the duplication of a gene on a chromosome. We see large-scale deletions, so entire genes deleting. Translocation, entire genes or groups of genes are moved from one chromosome to another. All right, we see there. Inversions, so some of the, or a part of the gene, okay, reverses its direction in the genome. Trinucleotide repeat expansions. Okay, and this actually occurs during DNA replication. This is actually the this kind of is the responsibility of Huntington's disease. All right, we see these repeats here, and these repeats obviously can cause an issue, or these loop out structures. Okay, this repetitive nature of DNA can cause an issue when there is protein synthesis involved here. The last two slides I want to talk about transposable elements. Right? Transposable elements, there's DNA transposons and RNA transposons. And these transposable elements are found in our genome. And what they have the ability to do is cut themselves out of our DNA and insert themselves into other areas of our DNA. Well, this could obviously be a problem. Okay? These jumping genes, so to speak. Okay? When they cut and paste themselves, let's say inside of a gene, that could definitely cause a mutation and an issue. RNA transposons act a very similar way to DNA transposons, but RNA transposons actually need a reverse transcriptase. It's an enzyme called reverse transcriptase to change that RNA transposon to DNA in order to insert itself into the DNA somewhere else in a new area. Okay, this is very similar to how HIV works. HIV has RNA. It needs a reverse transcriptase enzyme to convert its RNA to DNA so it can insert itself into our DNA. Okay. Again, these are transposable elements, DNA transposons and RNA transposons. You'll probably see this in matching. I would like you to still know about them, what they could potentially cause down the road if there was a cut and paste in somewhere along the gene for transcription. There could potentially be an issue there. Again, this would probably be seen in matching. I would like you guys to review these slides. The regulating gene expression is the one that I want you to know the most on right now, because that's going to be a big question on the test. I want you to know all 4 of those ways in which genes can be regulated. Again, the single nucleotide polymorphisms, the small scale mutations, I am going to ask you a question. You will need to transcribe and then translate DNA and then mRNA to a polypeptide sequence and tell me what type of mutation it was. Was it a frame shift? Was an entire sequence of amino acids changed? Or was it, let's say, a missense where there's just a single amino acid changed? Or was there no change at all? So that would be a silent mutation. So you would have to obviously do a little bit of transcribing and translating yourself and then determine what type of mutation it was. Please review these slides. When I see you in class, please please ask me questions that you have that you need answered. And again, we will review this before the test.